I'm a little older, and when I was in school, one of the emphasis in genetics classes was, oh, the gene was found. They just found a gene for this. They just found a gene for that. So when I was in school, finding genes, what gene encodes this protein, what gene does this, that was, quite an emphasis of research. Since then, however, a major focus has then been, how is the gene regulated? Because if you were to say, look at the genes of the human body. So here's, for example, cells of the esophagus and cells of the trachea. And then one even looks, here we have, epithelial cells lining the lumen of the esophagus. We have smooth muscle cells. We have cartilage cells. We have these epithelial cells which have cilia, while these do not. We realize, wait, they all have the same genes. All right, so whether you are a nerve cell in the human body, which is capable of conducting electricity and secreting neurotransmitters, or whether you're a muscle cell, or whether you're a liver cell, breaking down toxins in food, etc., all of the same genes are in the nucleus. So the same 46 chromosomes. And the same 26,000 or so human genes are in this nucleus as was in the nucleus of those cartilage cells, et cetera. So it's not just a question of having genes. It's a question of which ones you're using, how you regulate these genes, which will distinguish this white blood cell from this fibroblast, et cetera. As we study disease states, very often if you ask what is making this cell cancerous as opposed to this other cell, the answer would be the gene expression, all right, that a cancer cell shouldn't express a certain protein, and when it does, that's why it causes cancer, or it should express a certain protein. So sometimes there's no difference in expression between a cancer cell. But then sometimes there is, that this protein is expressed only a little bit in a normal cell, but at a higher level in a cancer cell. So a big question is not just then what genes does a cell have, but how does it regulate them? How is it expressing them? Well, before we get into all of the levels of human gene regulation, it should be stated that everything regulates their genes. Even viruses can, which aren't even alive. So the simplest genetic systems we have, those viruses do. If you, for example, were to consider coronavirus, if we look at how the genome is organized, so there is an open reading frame that starts with a start codon at this end, which makes one protein. Then there's another open reading frame. And these polyproteins, which are then cut into lots of little functional proteins, these are then produced first. It is only then later in the infection cycle that the other genes are then producing the S, E, M, and N proteins. In fact, the coronaviruses belong to an order of viruses called the nidovirales. Nido refers to nest. So for example, in Spanish, or I think Latin, nido means nest, that these genes are nested all in one end of the genome, all in the same order, but then we have genes which are active earlier in infection, like those which are going to reverse transcribe the DNA, fix errors, et cetera. They are made earlier. So viruses can regulate their genes, expressing some genes earlier in the infection cycle, and then some later. And then the same is true of bacteria. Now, I'd like to start with the topic of operons and just mention them because, once again, I'm old and I laugh at this with my students, but in all seriousness, I think one of the jobs that instructors have, certainly in genetics, my instructor certainly did, is they give you an idea of the passage of time. Like I often point out to my students, when I was in school, this wasn't known yet. This was discovered recently. We didn't know this. We thought this. We were wrong, etc. My instructors did. And certainly, I think in any topic in science, but certainly in genetics, the pace at which new knowledge is coming, it's amazing. And so you can say, someday my students will say, my instructor, when he was in school, they thought this, or this was the situation. And then my students may watch that, where if they get to become teachers, they'll tell their students, look at how much has been learned just in the decade or two that I've been teaching. So for example, I'm going to make this discussion of operons very short. That was not the situation when I was in school. Operon discussions went on forever. You could have a whole course. I took this one course once that so much of it was on operons. Why? Well, because back in the day, that's what was understood. All right, how are genes regulated? When I was in school, we understood some things about bacteria, but then eukaryotes was largely just, that would be how the chapter ended. Maybe someday we'll understand them, which are so much more complicated. And then I remember there would be then the chapter that would then have like the page or two at the end. Here's been a discovery or two from yeast, and so we're beginning to get insight into how eukaryotes regulate their genes. And then more was understood, so that it would be a chapter all by itself. And now, because genetics courses are still just one semester long, but we now learn so much more, this operon stuff, we just don't have time to go through it in any great detail, because there's so much cool stuff about, how human genes are regulated. So I think it's, that perspective of how quickly new knowledge is being amassed is important. So I always point that out to my students. So what is an operon? Well, in bacteria, and a couple of examples in lower eukaryotes, there are examples where genes share an on-off switch. All right, so there are regulatory elements of a gene which decide when you turn it on or when you turn it off. And in humans, all of our genes have their own separate on-off switch. Every gene has its own regulatory mechanisms. While in bacteria, you can find what are called operons. Well, gene number 1, gene number 2, and gene number 3 all share the same regulatory elements. So once again, this is a way of, let's say a bunch of genes are produced to make, a certain metabolite. Well, you could all then regulate them together. What are the parts of an operon? Well, two we already know, the genes to be transcribed, and then the promoter. The promoter is where RNA polymerase binds to start transcription. But then there are two new pieces. One, there is this section next to the promoter called an operator. The operator region is where a protein known as a repressor can bind. The repressor is made by this gene called the regulator that doesn't have to be nearby. It can be somewhere else in the genome. It simply then is making this repressor molecule, which will then bind to the operator. When the repressor molecule has bound to the operator, it will stop RNA polymerase from performing the transcription of the genes. So in other words, you can turn the operon off, all right, and not allow the transcription of the genes when the repressor molecule is bound to the operator. Why would you want to do that? Well, just very simply, to be efficient, all right? Organisms, well, they have to make it through their day. They can't waste energy. They're competing with other organisms. Organisms which use energy only for those necessary requirements, they're going to excel and do well and outcompete other organisms. Organisms which are wasting energy on unnecessary processes, they are not selected for. And so, for example, let's imagine here are the bunch of genes in the lac operon, which are involved in the metabolism of lactose. So if lactose is available, if it's a food source, well, breaking lactose down, that's an advantage, all right? And so here are genes which will help a bacterium break down lactose. And because they all share the same regulatory elements, they are known as the lac operon. There's the promoter, and the RNA polymerase can bind to the promoter, transcribe the genes, and then they will produce proteins involved in the metabolism of lactose. But it would be a waste of energy to transcribe these genes if there is no lactose. Why make enzymes to break a foodstuff down if it's not there? And so you want to regulate the lac operon so that it is only made when lactose is present. So under normal conditions, if lactose is not present, the regulator gene makes the repressor molecule, which then binds to the operator region and blocks transcription. Okay? And so under normal conditions, the lac operon is off, which is good. It makes sense not to make the genes to metabolize lactose. If there's no lactose, that would be wasteful. But this operon can then be turned on. So it is what is known as an inducible operon. So it begins off, but it can be turned on. And it can be turned on by lactose itself. Lactose is what's known as the inducer. When lactose is present, lactose can bind to the repressor molecule, and this will then block the repressor from binding the operator. So when lactose is present, it prevents the turning off of the operon. So lactose is an inducer. It can bind to the repressor molecule. This will inactivate it. It can't bind to the repressor, and so now the operon is on. So note, here's lactose. Lactose is now present. So it makes sense to produce the genes and proteins which will metabolize lactose when lactose is present. So this is an example of an inducible operon. It starts as off, but it can be induced. It can be turned on. Now, one of the things that I studied to far greater detail is all of these genes that you see then, they could have mutations, which would then affect the regulation of the lac operon, and this, allow the study of this system. So, for example, there can be a mutation in which the regulator gene makes a faulty repressor, a repressor that does not bind to the operator region. Therefore, if the repressor doesn't work, then this operon will always be on. It can't be turned off because the repressor doesn't bind there. And so that's one type of mutation. Other repressor molecules might not be able to bind lactose, the inducer. And so if lactose can't bind the repressor, it can't turn the repressor off. And so this would then be a super repressor, which would almost permanently inactivate the operons because it could not then be induced. And so one of the things that we could then go through are a variety of different mutations and talk about how they could affect the activity of this operon. So it turns out there are a lot of operons which are used in bacteria. I'd just like to give one more example. The tryptophan operon encodes genes necessary to make the amino acid tryptophan. Now, tryptophan is an amino acid that bacteria have to make, but it would be a waste of energy for the bacteria to make it if it's in the environment. Why make something that you can simply absorb as food? And so will the transcription of this operon occur? Well, it depends. Now, if this looks odd to you, the idea that the red is the DNA, and here we are, here's RNA polymerase performing transcription, and here we are doing translation. If that seems odd, it's because bacteria don't have a nucleus. The nucleus and the nuclear membrane separate transcription, which happens in the nucleus, with translation, which happens in the cytoplasm. Bacteria, they can occur together. And so here you can see translation occurring. And as translation begins, we get to a spot here where tryptophan is needed to make this short little peptide. Here's the tRNA for tryptophan. But if tryptophan isn't present, then the tRNA can't become charged. There's no tryptophan to stick to the tRNA, so the tRNA can't go here. So here the ribosome starts making a little peptide, but it can't go any further because there's no tryptophan. As it hits this, the pausing of the ribosome causes the mRNA to form this loop. This is what's called an anti-terminator loop. And when this loop forms, it allows the RNA polymerase to keep on going. That first ribosome dissociates, all right? A second one then forms, all right, at a second start codon. And now we make the genes which will produce tryptophan. So if you notice the advantage here, you want to be efficient. Tryptophan wasn't present. That's why that anti-terminator loop formed. And under the condition when tryptophan isn't present, the cell makes the operon, all right, so the operon is now on, which will then synthesize tryptophan. So you make tryptophan when you need it. When tryptophan isn't there, you make some. So this is a repressible operon. It starts out in the on state, but it can be turned off. Okay, it can be turned off when tryptophan is present. All right, because we'll see in the next video, when tryptophan is present, then those tRNAs for tryptophan can become charged, all right, and they can then bring tryptophan to the ribosome as it's performing translation. And this then allows the mRNA to form a new loop, a terminator loop. So see, notice there was tryptophan here, and this short peptide requires tryptophan. It's obviously present. And now, because the ribosome did not stall, what happens is a loop here will form called a termination loop. And this displaces RNA polymerase, so transcription will stop. So the cell does not transcribe any further for the genes to make tryptophan, because obviously tryptophan is present given the presence of tryptophan there. Okay, so there are so many other topics. Once again, when I was in school, week after week, we would still be talking about operons. The big point is, before we start getting into humans, is that number one, we find the regulation of genes even in things as simple as viruses and bacteria. Bacteria often have these things called operons, where there's one regulatory set of genes or regulatory elements for a set of genes. And there are both inducible operons, which are off but can be turned on, and repressible operons, which are on but can be turned off. With that, I'd like to now jump into eukaryotes, focusing on humans, and talk about how higher organisms regulate their genes. And so then this is, a key to complexity. I have brain cells, I have muscle cells, and they all have the same genes, all right? I'm old now, but I was a child once, and I was an embryo before that, all right? I was using different genes when I was an embryo than I am now. So how did that work? And the answer is because just because I have a gene doesn't mean I'm using it. We regulate our genes. Certain genes are active, at certain times in our life, or active in, say, nerve cells, but not in muscle cells, etc. So we regulate genes. Just because we have 3 genes doesn't mean we have to use all 3 of them. All right, if you go to a brain cell, it might just be using gene 1. If you go to a liver cell, gene 2. If you go to a muscle cell, gene 3. If we consider fetal development, you might have used gene 1. All right, after a meal, you might use gene 3. And when you're fasting, you might use gene 2. When you've cut yourself and you have to heal, you might now activate gene 2. So that's the key to our complexity, not just the genes that we have, but how we regulate these genes. How do we regulate these genes? Well, this is complicated, and it's so complicated that what I'd like to do is to give one, I don't know what just happened, give kind of this lecture as an overview, hitting the highlights, but then go back in future lectures and then take some of these topics and deal with it in greater detail. So there's a value to just starting at the beginning and going through to the end, but that would also then sell a couple of topics short. So I'd like to then revisit those. So humans and other eukaryotes, well, let's just talk about humans. Humans can regulate their genes at multiple levels. We can regulate genes at the transcription level. Do we transcribe a gene or not? At the post-transcription level, at the translation level, and at the post-translation level. So there's transcriptional regulation, post-transcriptional regulation, translational regulation, post-translational regulation. The major one, the primary way in which we regulate our genes is the regulation of transcription. So I'd like to begin there. So how do we regulate which genes get transcribed or not? And there are a couple of ways. The first way is we regulate how tightly packed our DNA is, all right? So when we look inside the nucleus, we see chromatin, but some chromatin is in the form of what's called heterochromatin. It is very tightly wound. Heterochromatin is not transcribed. It is not active. It is only the unwound DNA, the euchromatin, which is active. Now, an example that I often use in my class is I say, when we think of, mitosis and we're getting ready, to divide the cell. Obviously, like if you were to think of your house, you've got stuff all over the place, but you can find stuff. You can find your, genetics notes. You can find, the t-shirt you want to wear, et cetera. You can find everything. But if you're going to move, if you're going to do mitosis, you pack everything up in boxes. All right, so now it can be moved, but now it's not accessible anymore. So, earlier in this class, you might have, when you talk about, prophase, where the DNA is condensed, we know that unwound DNA is the form we find when it's active, and we can use it. When it's not going to be used to make protein, that's when we tightly pack it. Well, we can see that even in a nucleus at rest, that some DNA is more tightly packed than others. So there's a difference between heterochromatin and when it's tightly packed and not being used, and euchromatin, which is unwound and can be used. And so if you ask, how do we regulate our genes? How do we say use gene number 1, but not use genes 2 and 3? And one of the answers can be, well, we make genes 2 and 3 heterochromatin. They are more tightly wound. And by being tightly wound, they're going to be inactivated. And we can change which ones we wind versus loosen. And so there are areas which are always heterochromatin. They're what's called constitutive heterochromatin. So you're not really going to express any genes here at all. But then there can be facultative heterochromatin where you can wind it up and inactivate it for a bit, but then you can then unwind it as well. So notice in this constitutive heterochromatin, no gene expression is occurring here, but in facultative heterochromatin, we can express it for a while, maybe during childhood, or maybe after a meal, or maybe after I've cut myself. And then later, when appropriate, to then make it heterochromatin so that we turn it off and we don't use these genes. So for example, when it comes to cell division, if cells were always producing cell division proteins, that would promote cancer. So we want to make proteins when appropriate, but then turn them off when inappropriate. Otherwise, we couldn't regulate our cell cycles and we would develop cancer. So here, as you see, we have all of this euchromatin. It is unwound, and so those genes are accessible. But if we wanted to prepare for cell division, if we wanted to inactivate proteins or genes, we could then form heterochromatin. And then that's one way of regulating transcription, all right? So one of the things that you can do is, and some people would say, study the DNA in Drosophila salivary glands, or the chromosomes in, say, amphibian eggs. And you would notice, wow, here's an area of heterochromatin, no gene expression is occurring there. Oh, wait, look, an area just became euchromatin, all right? And so this area that was heterochromatin now became euchromatin, and so now a gene here could be expressed. It's accessible. RNA polymerase could bind to start transcription. And so you could see, say, in Drosophila salivary chromosomes, that there would be a change over time, the areas which were heterochromatin versus euchromatin. So, when the area goes from heterochromatin to euchromatin, you're potentially turning the gene on because the gene could now be expressed. If you did the reverse, if you took what was euchromatin and unwound, and then you made it tightly wound heterochromatin, that would be a way of turning the gene off, okay? And once again, we regulate transcription in a couple of different ways. And the oops, I'm sorry, should have ended that. The regulation of whether an area is unwound euchromatin, which can be activated, or tightly wound heterochromatin, which is inactivated. This is one of those ways. Now, two of the big ways that humans utilize this is if you were to visit, one cell in the body versus another one, you might see, oh, here we have, I don't know, a liver cell. Oh, I'm sorry, I actually wrote it. Yeah. So let's say here's a liver cell, a hepatocyte, whereas here's a fibroblast, say, in connective tissue. All right. Well, you might say, oh, look what happens here. In the liver cell, the gene, or this area of the chromosome, becomes tightly wound heterochromatin, while in the fibroblast, the area becomes unwound euchromatin. And so here's one of the ways in which we would regulate genes, in that we can do it by cell type. All right, all of these cells have the same genes, but what's happening is the liver cell is turning a gene off while the fibroblast is turning a gene on. This is one of the things that could make a liver cell different from a fibroblast. Now, this can occur in us right now, but then also it could occur during fetal development. Right, so there might be some genes which you would only use in fetal development. So during at some stage of fetal development, you would turn it on by making it euchromatin, and afterwards you would turn it off by making it heterochromatin again. A very famous example of this, and one of the reasons it's famous is because this was one of the first ones discovered, is it turns out the human genome has a number of different genes for globin. And there are two major clusters of the globins, the alpha cluster and the beta cluster. So not only is beta hemoglobin there, but there are a couple of genes located in the same cluster with the beta hemoglobin. So beta hemoglobin is here. But there is also an epsilon, a gamma G and gamma A hemoglobin, an eta gene, a delta gene. And so what we observe is that, I'm sorry, I can't access, this gets in the way. But what we see is that these different genes in this cluster change as to whether they are heterochromatin or euchromatin. So notice that the epsilon globin, it becomes euchromatin in the first trimester and makes protein. But then it becomes heterochromatin and is then not used again. In the second to third trimesters, the gamma globins become unwound euchromatin, which then get used, and then but then they become inactivated. And then later, after birth, the delta and beta globin genes become euchromatin before being inactivated. So here you can see that over the course of development, there is a change as to which genes are euchromatin versus heterochromatin as a way of regulating the genes. And elsewhere, we can see this with cell type. Now, one of the interesting things that can happen is when something becomes heterochromatin, it can it can bind intermediate proteins known as Lamin filaments. Just as there's a cytoskeleton of the cytoplasm, there is a nucleoskeleton of the nucleus. And these Lamin proteins are giving the nucleus its structure. Nuclear Lamin proteins preferentially bind to tightly wound heterochromatin. So proteins bind the sections of DNA which have been turned off, and the lamins can then migrate these areas of heterochromatin to the nuclear membrane. So gene areas which are off can now be physically located in a different region than the genes which are on, because once it forms heterochromatin, the Lamin proteins can move it over here, as opposed to the euchromatin, which could now be more in the center. So here's another aspect to the regulation of the genes. Once something is off, it can physically now be moved to a different area of the nucleus and have proteins bound to it. Mutations that affect Lamin proteins can be a cause of cancer, giving you an idea that this is an important factor in the regulation of genes. Okay, so most of the regulation of genes which occurs at the level of transcription. And there are actually a couple of different ways. One way in which transcription can be regulated is by determining whether an area is euchromatin or heterochromatin. The second way that transcription can be regulated is the presence of transcription factors. This is one of those things I mentioned earlier where this is such an important topic that what I'm about to say is a quick overview of it, but there's far more details that could be given in its own separate lecture. So let me give a quick overview. So in bacteria, as you saw in some of the operons, RNA polymerase can sometimes bind to DNA and then just start transcription. That does not happen in eukaryotes. RNA polymerase cannot bind DNA to start transcription unless there are other proteins bound first. All of our genes, all of them start in the off position. So gene number 1 is off and no transcription will occur. RNA polymerase cannot bind unless certain proteins bind to the beginning of the gene and the regulatory elements of the gene. These are called transcription factors. So proteins have to bind DNA in order for transcription to occur, these transcription factors. Without these blue transcription factors, the blue gene does not get transcribed. Now, obviously, this then gives the cell the ability to regulate transcription because it can choose which then transcription factors are made. If the cell wants to express the red genes, then it can make the red transcription factors, or the green genes, it can make the green transcription factors. And so proteins need to stick to DNA in order for a gene to be turned on and transcribed. And so the proteins which do this are known as transcription factors. Do human proteins, or does the human genome have transcription factors? Yes, hundreds and hundreds and hundreds of them. There are some transcription factors which are general and are needed for, say, transcription in general. But then there are transcription factors which are needed then for specific genes. And there are so many hundreds of these transcription factors, often in what are called gene families. So not to get into it, here, but the way that more genes have appeared over time is through gene duplication with then subsequent mutations changing them. So, for example, there are transcription factors known as zinc finger transcription factors. Once upon a time, there was only one or a couple of these different genes, but then there were accidental duplications and there was more and more. And because they changed a little bit, now we have a whole bunch of what's called a gene family, a bunch of genes all descended from the same ancestral genes. There's lots of them. And if you recall back to the cancer lectures, one of the parts of the cell cycle is at some point messages go into the nucleus where they stick to DNA and now change the transcription of genes. So as you tell a cell, hey, you were resting, but now it's time for you to divide. Well, you have to change the messages. So you have to then turn on genes which had been off. So messages then have to reach the nucleus, bind to DNA, and then turn on genes. When we think of puberty, at first, puberty is a little odd because you think, wait a minute, why is the body different in late teen years than it was in childhood? There are no new genes, right? We don't get new genes. So why are there differences? And the answer is because now there are differences in what genes are being used. And that's because estrogen and its receptor, when they bind together, they stick to DNA, all right? They are transcription factors. Testosterone and its receptor stick to DNA. And so we make different genes after puberty, once estrogen and testosterone have stuck to DNA, than we do prior to puberty. And so transcription factors are incredibly important when we talk about cancer, for example, because a number of transcription factors are proto-oncogenes. They can cause cancer. Another important topic would be all of the gene families of transcription factors. The helix turn helix transcription factors, the homeodomain transcription factors. So from that standpoint, that's important. And then obviously these are important in embryologic development. Now, once again, this is such a huge topic and I prefer not to, run down every avenue here. Let me just wrap up with a couple of things. So there aren't that many genes on the Y chromosome. One of the genes is what's called SRY. It's the testes determining factor. This is a major determinant of the male gender. Males have all of the genes that females have, all right? And females have all of the genes that males have except one, SRY. So if you look at the differences between men and women, why? They've got all the same genes. Men have all of the genes that women have. Women have all of the genes that men have except for one. Why the differences? Well, because that SRY gene, it codes for a transcription factor that then turns on other genes like FGF9 and SOX9. They are transcription factors. I'm sorry, SOX9 is. And then they then bind to DNA and turn on more genes. And so a lot of these transcription factors are really important in embryological development, because an embryo starts as a ball of cells, right? But somewhere you have to say, make a brain here. And if you're the brain, make the forebrain here as opposed to the hindbrain there, or make liver over here. Well, how do you know in a ball of cells what you should do? Well, you have to turn on the right genes. You have to turn on the brain genes or turn on the liver genes, et cetera. And so transcription factors go very quickly into the regulation of embryonic development. And, as we talk about the, there's so many aspects. So, but one of them could be, at some point, our ancestors stopped developing into regular old apes and started developing into early humans. How did that happen? Well, what transcription factors modified human development in a different way? And we can study those, including the transcription factors which seem to have been important in language development, like FOXP2. And so I'm going to go back. I have a lot of videos on the different families of transcription factors, how they work in embryological development. So important. But let me now go to a third way that transcription can be regulated. Now, we know that cytosines are one of the nucleotides. But one of the interesting things is cytosine can have a methyl group attached to it. If you've studied organic chemistry, you know that methane is CH4. A methyl group is a CH3 group that can be then attached to another molecule. So here you see that methyl groups have been attached to cytosines in gene 2 and gene 3. That is one of the things that turned them off. And actually, this is an early stage which could then lead to the promotion of heterochromatin. So once we add methyl groups, to cytosines, this could then make it likely that they then become heterochromatin and are turned off. Now, this is another topic which could then just get into the much broader topic of epigenetics. But once again, I think the best thing would be just for me to stick with this, one theme, how we regulate genes, and then make epigenetics its own separate discussion. And so here you can see that the difference between this active gene and this inactive gene is the fact that we have put methyl groups on the cytosines of the inactive gene. And as we'll see when we discuss epigenetics, that this can then change the packing of the DNA around proteins known as histones, and these methyl groups and then also acetyl groups being attached to histones can then change the activity of a gene without changing the sequence. So epigenetics studies that. Now, I'm going to make this topic in two parts because I think that would just affect the flow easier. So let me wrap up then part one. How do humans regulate their genes, decide which proteins are on, off, et cetera? There are lots of ways, but the most important ones act on transcription. All right, because obviously if you're not going to use a gene, or if you're not going to use its protein, then if you transcribe it, you've wasted energy. So obviously the earlier in the process you can act, the more efficient you are. Don't transcribe the genes that you don't intend on using. So transcriptional control is the major way in which we regulate our genes. And there are three ways that transcription is regulated. One, we can control whether an area is tightly bound heterochromatin, which won't be transcribed, or loosely bound euchromatin, which can be. Two, we can choose to make transcription factors which bind to the beginning of a gene to activate its transcription or not. If we don't make the right transcription factors, a gene will not be transcribed. And then three, if methyl groups are attached to cytosine nucleotides, then the gene is more likely to be off. These are ways of regulating transcription, and transcriptional regulation is the most important step in the regulation of human genes. But as will then be described in the next video, our regulation of genes doesn't stop there, because we can still regulate genes after transcription, what's called post-transcriptional regulation. We can regulate the process of translation. We can then also regulate proteins after they've been translated, as I'll discuss in the next video.